is descriptive. The testing on this chapter is going to be concept related, as opposed to actually asking real questions about big time content. So think about the concepts as we approach them, and we'll just work through them. Next Monday, we will do the sample exam, so you'll get a better idea of the kind of stuff that you need to think about. Let's start off with the concept of valence. We've talked about this before. In its ground state, its most stable state, elements will form a number of covalent bonds. Oxygen tends to form two. We have two unshared pairs of electrons, so it has an octet. Nitrogen will form three. The geometry here is pyramidal, but it's actually tetrahedral for electron pair if you count the unshared pair. Boron only has 6 electrons, poor guy, and it's a very strong Lewis acid, which means it really, really wants another pair of electrons. Carbon is 4. Chapter on organic, we're dealing with carbon. We want 4 bonds to our carbon. They can be single bonds, double bonds, triple bonds, but there have to be a total of 4. Now, if we look at carbon in our periodic table, it's in the second period atomic number of 6. So, or right over here, if we were to draw the atomic structure, we would say because it's in the second period, the first is full. So it has 2 electrons. First only has an S, so we have 2 electrons in our 1s. Now carbon's over here, so on the second period we filled up the 2s as well. Now we have a total of 6 electrons, so we're going to be 2 here, then 1, 2, 3, and 4. So that means the p orbitals have to have 2. Now, as you recall, these come in with the same spin, one at a time. Now, as you look at this, you might think that, well, gee, how does carbon make its 4 bonds? Well, in order to make 4 equivalent bonds in carbon, we have to take one of these s electrons and promote it from the s orbital up to this empty p orbital. This is called hybridization. So here we start off with 2 in the 2s, 2 in the 2p, and we're simply going to move one of these guys here. So we have 4 electrons that we can make bonds with. This is how we get a valence of 4 from carbon. The simplest carbon compound is methane. Methane has 1 carbon. It has 4 hydrogens. It is tetrahedral. That means that all of the bond angles here are 109.5. They're all equivalent. They're all exactly the same. All the bond lengths are the same. If you think about what a tetrahedron looks like, and this isn't something that they teach in kindergarten, they should, the carbon here is kind of suspended in the middle of this little pyramid. So it's suspended up here, so it's right smack in the middle. All these are equal, and again, all the bonds are exactly the same. So, bottom line, whenever you look at a carbon compound, it's going to have 4 bonds, and for right now, we're going to have only single bonds that were made from 1s and 3p orbitals. So we call this an sp3 hybrid. It's a hybrid because we took 1 electron, moved it up, and made 4 equivalent orbitals. hybridized orbitals. We'll use the word hybrid as we deal with other types of carbon compounds as well. Now, as we go to build carbon compounds, we can build them using only sp3 carbons. For example, if we have 3 carbons in a row, each of these carbons has 4 bonds, so that's good. 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. That's good. That's what it's supposed to be. If you realize that each of these carbons is a tetrahedron, then that implies a geometry to this molecule. The geometry looks something like this. Once again, all of these bond angles everywhere are 109. All the bond lengths, well, CH bond lengths and CC differ slightly, but they're basically all the same. This describes what we call in organic chemistry a functional group. Now we'll do lots of functional group stuff later on, but the neat thing about a functional group is that all carbon compounds that belong to that particular group will have similar reactivity. This allows you, because there's so many possible carbon compounds, an infinite number, so many possible carbon compounds, it allows you to look at a compound, identify the functional group, and then you'll know the classes of reactions that this compound will undergo. That's how you can do organic chemistry. This is an alkane. An alkane only has sp3 carbons connected together. Now we've connected them here in a straight chain. We could pull off one of these hydrogens and stick a CH3 on there. If we do that, the compound looks like this. So here's our straight chain, and here's the CH3 that we just stuck on. This is still an alkane because all carbons are sp3. Any questions on the concept? This is also called a branched chain alkane. Here's our main chain, and here's our branch. You'll note that this thing looks like a zigzag. And we will use that zigzag later on as we learn to draw simple organic molecules. If you add up the numbers of carbons and hydrogens in this compound and in this compound, here we have our 1, 2, 3, 4 carbon main chain, here's a 3 carbon chain, a 1 carbon side chain. We wind up with a formula where we can predict the number of hydrogens in the molecule if we know the number of carbons. The formula is simply Cn, where N is the number of carbons, and we have 2N plus 2 hydrogens. So here we have 3 hydrogens, which means we have 2N, so that's 6 plus 2 more, 8 hydrogens. Over here we'll have 1, 2, 3, 4, 5. So 2N is 10 plus 2 more. This is C5H12. Whenever you have an alkane, the general formula will be Cn H2N plus 2. That's how you know it's a simple alkane. Now, when we talk about alkanes, we need some system of nomenclature so we know what the heck we're talking about. Again, there are so many possible organic compounds. We're going to do this by looking at our molecule, finding the longest continuous chain. We'll call this the parent. Okay? The longest continuous chain will be the parent. Then we're going to look at the number of carbons in that longest continuous chain.
Okay.
When we do that, we can assign a name. One carbon is methane. We've seen that. 2 carbons is F. That's our prefix. We add A and E to the end of the prefix so that we know it's an alkane. A and E is a suffix that indicates we're dealing with an alkane. So here our prefix is F. So this is ethane with 2 carbons. Prop, bute, pent, hex, hept, oct, and then they get bigger. You should probably appreciate the lower ones for sure, up to maybe oct or so. Once again, the set of prefixes go het, f, prop, bute, pent, hex, hept, and oct. A and E simply says it's an octane. If I say we have octane, that means we have 8 carbons strung in a row. Now we can also have what are called alkyl groups. Remember we were taking this chain and we stuck a CH3 off the side. Well, that's a 1 carbon group, isn't it, that we stuck off the side. From our previous nomenclature, we know that CH3 is going to be a meth. But if it's a side chain, we're going to call it a methyl. YL on the end, replacing ane. And that simply tells you that we're dealing with a side chain. 2 carbons we said was F. So if we have a 2 carbon side chain, it would be an FO. Ethyl. This type of nomenclature goes throughout other functional groups as well. This is methane. We pull the hydrogen off, stick it on as a CH3 group on a chain. We call it a methyl. If we stick on an OH, that's going to be an alcohol. We'll see that later. Alcohol, the name ends in OL, so this would be meth for 1 carbon, and because there's only single bonds, and OL because it's an alcohol. Here we have an amino group in here, a nitrogen. Once again, meth, methyl, amine. Nomenclature for more advanced functional groups is all based on the main chain, which, meth, eth, prop, and then side chains, methyl, ethyl, propyl, whatever. It's actually a very simple, systematic name, a way for nomenclature. Once again, we have an alkane with 1 carbon. We call that methane. If it's a side chain, we call it methyl. 2 carbons, ethane, ethyl, propane, propyl, butane, butyl, and pentane, pentyl, and on and on and on. That's how you name side chains on organic compounds. Now, I've said that organic compounds, you can have billions, billions, infinite number of them. That's because carbon is really capable of forming all sorts of configurations, all sorts of bonds, whatever. Here we have a simple alkane. We have 1, 2, 3, 4, 5 carbons, so it's a pent. It will be a pentane because it's an alkane. We have 1 CH3 side group, so we'll call this a methyl group on a pentane. This would be a 2-methyl pentane. Here we've taken the carbons and we put them in a ring. Now if you look at the way this ring looks, is built with zigzags up and down, isn't it? That's because, remember, each of these is a tetrahedral carbon. And all the angles have to be the same at about 109.5. So this is a derivative of an alkane. It's called a cycloalkane because we have a ring. Over here we have a more complex structure. Here we have a 1, 2, 3, 4, 5, 6, 7 carbon ring with another ring built in that's 1, 2, 3, 4, 5, 6. So this is a bicyclic alkane. The structures of carbon compounds can be very, very complex. Not only that, but carbon can form bonds to oxygen, nitrogen, sulfur, and darn near anything else. So we can stick in other atoms into these. This is glucose. Glucose has a 6-membered ring right here. It has OH groups all around here. An oxygen bridges in the ring, and we have a CH2OH sticking up here. This is morphine. Here we have a combination of double and single bonds. We have a nitrogen in our ring here, another ring attached out this way, an OH, an oxygen bridging 2 carbons, another OH. Again, the structural variabilities are just amazing. That's why organic chemistry is probably the neatest thing you'd ever do in your life. Now, I really like organic, and that's because it really can be a very neat subject. All right, so we have a compound. How do we show this compound? If we want to use molecular models, and we use a mixture of all of these in lecture, we can do what are called ball and stick. With ball and stick, you show the atoms as little balls. You show the bonds as sticks. That's the name. So this is a 4-carbon chain, CH3, CH3, and 2 CH2s. Something called a Drying model. Drying model is just a ball and stick without balls. So it's just stick. We look at this and we would say CH3, CH2, CH2, CH3. Probably the best representation is to use a space-filling model. Problem with space filling, even though it looks beautiful and it tells you just what the molecule looks like, it's not real useful because you can't see the bonding very well. If you had multiple bonds somewhere in here, you would never see them in a space-filling model. Plus, you can't really draw one. Now, if we're going to do a simple organic molecule, we could represent that using a simple molecular formula. We have 4 carbons and 2, 3, 6, 8, 10 hydrogens. That's our molecular formula. But the molecular formula tells you nothing about the connectivity of the atoms. For that, we would use a condensed structure. When we write a condensed structure, we try to emulate what this stuff really looks like. We start with a CH3. We attach that to a CH2, another CH2, and another CH3. When you read a condensed formula, you're supposed to just understand that the carbons are connected together and that the hydrogens are dangling off so that everything has 4 bonds. We can also use what's called a Kekulé structure. Kekulé structure very clearly shows our carbon chain, shows all the hydrogens duly attached. Problem is that you get really, really tired of drawing little lines and little hydrogens. Therefore, the line drawing or the structural drawing is typically what you will see. Now, when you see a line drawing, you are expected to know that if this line goes off and ends, just stops, that this represents a carbon with 3 hydrogens. If we have a vertex in our drawing, this represents a carbon with 2 hydrogens. So our CH3, CH2, CH2, and another CH3. You're just expected to look at this and to understand that. Every carbon will have the appropriate number of hydrogens in this type of a drawing. This is important because most of the structures that we'll see will be represented as line drawings or modified line drawings. You don't show the hydrogens, but every carbon has as many as it needs to get to 4. So, this is a strict line drawing, 4-carbon chain, and again we do it as a zigzag. Why? Because remember, that's what it really looks like, doesn't it? They're all 109 degrees, so it goes up and down, up and down. We have a CH3 on both ends and 2 CH2s. Now, quite often people that write textbooks or do lectures or whatever will try to help you out to make sure that you understand what this really means. So you could see this written this way as well. Sometimes you'll stick the CH3 groups on the end. Now that's just designed to help you. That's good. However, if you look at this, some people would look at that and say, hey, this looks like this carbon is bonded to the hydrogen. Well, that's not right. Therefore, sometimes you can simply reverse this and draw it this way. This way we have an H3C, clearly shows our carbon-carbon bonds and the hydrogen simply attached. So you'll see this represented all ways. Just remember, when you see this, truncated line is CH3, a vertex is always a CH2. Now there are a couple other ways that we represent. This is a bit of a line drawing. This is pretty much one-dimensional. A lot of organic chemistry depends upon geometry. We're very concerned with the geometry of our compounds. So sometimes it's useful to be able to see this more in a 3D perspective. So, if you took this guy and you turned it so that this carbon came closer to you and this one went away, you would get what we call a sawhorse projection. This is our front carbon here and our back carbon there. In a sawhorse, we'll put in the hydrogens, probably not the carbons. So it actually, if you flip this upside down, it would look like a real sawhorse, but that's where it comes from. And this is useful if you want to look at the orientation of groups relative to each other. Finally, there's another thing you can do with a sawhorse, and that is to turn it all the way so that you're looking straight down this angle here. This is our front carbon, that's our back. This is called a Newman projection. This is our front carbon here, right there. Just to make sure we know who's in back, we take the back carbon and we make it a big circle. so we know that's in back. Our back carbon has one CH3, one methyl group, and two hydrogens. One methyl and two hydrogens. Our front one does the same, just like that. And as you look at this, this is useful because you could tell that the methyl groups here are opposite from each other, aren't they? And that's important. So that's why you do these different types of projections. Now this next slide is a little movie that simply shows the process of going from this to this to this, just to appreciate it. So let's take a look at it. This is a Dryden model. Here's our front carbon, here's our back. As we begin the rotation, we're going to move this towards us and that away. Continuing all the way, this will be front, and this guy is the back. Let's watch this one more time if we can make it work. Maybe not. So we rotate it so this is in front. Then this guy is going to be the back, and we'll make it big.
So this is from the back we're seeing.
So we're looking straight from the front. That's a front carbon. Big guy is the back. So we'll see how to deal with these in just a moment. Let's go ahead and do a simple exercise. This should be on your sheet. Let's take these structures, a variety of structures, and convert them into simple line drawings. No, thanks.
Oh, Lord, she's gonna be bouncing off the wall.
All right, so we look at our first thing here. Remember, the first thing you want to do is find your longest chain. This is the condensed structure. This tells us we have a 1, 2, this is a side chain. So 1, 2, 3, 4, 5 carbon chain. First thing you would do is take and draw your zigzag for 5 carbons. So our zigzag will start up, go up, down, up, down, up, down, and it'll look like that. Now, we've numbered this guy carbon number 1. We'll understand why in a few minutes. But on carbon number 2, instead of hydrogens, we have 2 methyl groups, don't we? Remember, a methyl group in a line drawing is just a line. So on carbon 2 here, we have 2 methyl groups. We can simply show them this way. And that would be an acceptable line drawing. Our next structure here, we have 1, 2, 3, 4, 5, 6 carbons in a ring. Remember, that's a cycloalkane. So what we have to do is simply reproduce the zigzag for the bonds for all these carbons. These are all CH2s, so they're just going to be simple vertices. So how would you draw this? It would look like that. 6 carbons in a ring. Every vertex is a simple CH2. This molecule is called cyclohexane, hex for 6. And this is a particular conformation. This is chair cyclohexane. If you have a good imagination, you can imagine this looks like a little lawn chair or something, and you're sitting back, putting your head here, your feet here, just having a nice time. Now our next one is a Newman projection. We have to unwind it. We can do that though. Let's just look at this. Our longest chain here is going to be 1, 2, 3 as our front carbon. Then 4 is our back, 5, and 6. So it's a 6-carbon chain. On carbon number 1, 2, 3, we have a methyl group. On carbon 4, we have a methyl group. So we need a 6-carbon zigzag with 2 methyl groups. And it would look like that. Now, just in case that wasn't what you drew, let's try it again. Let's unwind it step by step. I'm going to take this guy and go backwards, convert it to sawhorse. So this was the answer. If we draw this as a sawhorse, this will be front, that will be back. So we would start off with 1, 2, 3 carbons, that's these. 1, 2, 3. We go back to number 4, that's him. And then we go up for 2 more, 5 and 6. 6-carbon chain, methyl group on carbon 1, 2, 3, and 4. 1, 2, 3, and 4.
I'm not ready.
Now, you know what? If we draw this compound this way, if I asked you to do this and you did this, I would be just thrilled as I could be. But there's a fact about carbon-carbon single bonds that we need to address. And that is that whenever you have a single bond, the carbons can rotate around it roughly 10 to the 6 times per second. So very, very fast. So if we look at this structure here, so we have a 1, 2, 3, 4, 5 carbon chain with an ethyl group. And we realize that all of these bonds are rotating 1,000,000 times a second. Then any way you draw it is going to work. So instead of drawing it this way, you could draw it this way. This is 1, 2, 3, 4, 5, 6 with a 2-carbon side chain. Exactly the same molecule. They just rotate so quickly that you can't tell them apart. These are called conformational isomers, and they only differ by the way you draw them because of rotation. In reality, they interchange so quickly that we don't typically bother with them. We'll see that there is a difference, but we typically don't bother with them. Any way you draw it will work. As an example of this, this is a pentane on the next slide doing gymnastics. Okay? So this guy is rotating about all these bonds, it's rotating, vibrating, doing all this stuff. Any representation that you drew that looked like that would work. Another way to look at it. Here we have a 1, 2, 3, 4, 5 carbon chain with a methyl group. Here we have a 1, 2, 3, 4, 5 carbon chain and a methyl group in a slightly different position. Here our chain is 1, 2, 3, 4, 5, and here's our methyl group. These are all exactly the same compound. All right. Well, carbon compounds can get to be very, very complicated. And if we're going to try to describe a carbon compound and give it a name, we need to have a systematic way to do that. The International Union of Pure and Applied Chemistry, IUPAC, has come up with a very extensive set of rules that allows you to take virtually any compound you can imagine and name it. It's all based on the number of carbons in the longest chain. So if we look here, we have 1, 2, 3, 4, 5, 6 carbons. And this is our longest chain here. Our longest chain here would go 1, 2, 3, 4, 5, 6. Let's see, what's longest here? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7. Oh, I think 7 wins. Now again, all of these possible structures are interchanging so quickly. It doesn't matter how you draw them. But you want to look at whatever structure you have and identify the longest continuous chain. This is going to be the parent. Let's look at this. Take this compound, identify the longest continuous chain. Well, if you just follow your zigzag, you go 1, 2, 3, 4, 5, 6, 7, right? We could do 1, 2, 3, 4, 5, 6, 7 that way. Or 1, 2, 3, 4, 5, 6, 7 that way. So one possibility would be to do this. We could do 1, 2, 3, 4, 5, 6, 7 that way. Now if you look at these two chains, you want to see how many side chains they have. This one has a methyl group, a CH3, and a 3-carbon chain that's a propyl group also. Turns out this one also has a methyl group and a propyl. So these are identical. These are just different ways of drawing the same compound. Now as we were doing this, we also came up with a third way, and that was to go down this way, like that. Turns out that as far as IUPAC goes, this is bad and this is good. Why is that? Because you're looking for the greatest number of substituents. This has 2 branches, this only has 1. So that's the rule. The other rule is numbering. You're going to number this thing so you wind up with the lowest number sequence. The sequence isn't an average or anything like that. It's a sequence. And we'll see examples of that as we go. So let's go ahead and look at a different compound. Oh, our numbering, we would number here as 1. That gives us 2 as our first substituent. Now this, our first substituent, would be at 4, and there's only 1 substituent, and here we have 2. All right, take this guy. And step 1, let's find the longest continuous chain with the greatest number of substituents. As I see it, we could do something like 1, 2, 3, 4, 5, 6, 7, 8 carbons in a row. If we started here, we would do 1, 2, 3, 4, 5, 6, 7, 8. but that would have different numbers of side chains. So I think probably that is going to be our best parent chain. We're going to let this guy be carbon 1, so our 2 methyl groups here can be on carbon 2. That's better. That's going to put out here on carbon 1, 2, 3, 4. There's 3 carbon chain, that's a propyl. And on carbon 6, we have a 1 carbon, that's another methyl. So step 1, you identify your chains. Here we have 3 methyl groups. So we're going to call that a trimethyl. And we're going to have to use numbers to tell us where they are. As I look at this, I would say I have methyl groups at carbon 2, 2, and 6. At carbon 4, I have a propyl. Now our main chain is 8 carbons, right? So that's an octane. It's an octane with 2,2,6-trimethyl and 4 propyl. So let's write a name. The name is going to be based on octane. We're going to arrange these alphabetically. We don't care about the multipliers, so methyl comes before propyl. So we're going to start by calling it 2,2,6-trimethyl. We'll put a dash in and say there's a 4-propyl in there too. And this is all on an octane. 2,2,6-trimethyl, 4-propyl, octane. So you figure out what you got. You figure out your longest chain, octane, right there. You figure out what's attached. You put them in alphabetically.
Oh my God, I can't even spell it
Now,
right. It
looking
is
at
right
multipliers. And you don't leave any spaces. No spaces at all. Simply commas and dashes.
No spaces?
Nope. Commas and dashes.
Oh my God.
So let's do another one here.
Of course.
If we look at this
The
guy,
first part is scary.
compare this to what we just had. Here at carbon 6 we had a methyl group, right?
I don't understand none of this.
What I'm going to do is substitute a halogen here. So we wind up at carbon 6 with a chlorine. So you would look at this molecule and say, number 1, how many carbons in your longest chain? Well, it's an octane. There are 8 of them. Now what's attached? In carbon number 2, we still have 2 methyl groups, so that's a dimethyl. In carbon 4, we still have a propyl. And in carbon 6, we have a fluoro. So we string it together. And we say, alphabetically, 6-fluoro, dash, 2,2-dimethyl, dash, 4-propyl, octane. All one word.
What do you mean by alphabetical? Because I don't understand.
Alphabetical? Fluorine, methyl, propyl. Alphabet. A, B, C, D, E, F, G. All right, let's combine 2 tasks here and name these guys. This one's going to be simple. This one we have to unwind, don't we? So step 1. Take the Newman and unwind it and draw a sawhorse. Remember, this is in front and the big guy is in back. Well, our process of unwinding. This is our front carbon, with the CH3 down here. Our back carbon has the methyl group and the ethyl. Okay? So that's what it looks like in Isomers. Now step 2. Look at this and identify your longest chain. What's the longest chain of carbon atoms? Well, 1, 2, 3, 4, 5. Five carbons in a row. And number 3, which carbon, which end of this chain we're going to call number 1? Well, we want the lowest number for our substituent, so we could do 1, 2, 3, or 1, 2, 3, doesn't matter. So we'll just call that 1 for the heck of it. All right, so what's our parent? We have 5 carbons. 5 is pent. So what's a pentane? Now what's attached to our pentane? A methyl group. And where is it attached? Carbon 3. 3-methyl pentane.
Can you show me the parent code?
Here's our parent, our longest chain. 1, 2, 3, 4, 5. That's a pentane.
Starting at this CH2, CH3 is where you
Yeah,
count?
sorry, I started here. 1,
One,
2,
two.
3, 4, 5.
2, 3, 4. Okay.
Now our next one is really simple, isn't it? Because we have a parent chain, it's going to be 1, 2, 3, 4. Doesn't matter which way you go here. See these little bonds? This is a wedgie bond. This is a wedgie going back. That actually indicates in 3D whether this group is coming towards you or away. One of the big things in organic chemistry is stereochemistry, and that has to do with the orientation in space. So we can use wedgies to show that. This one is coming towards us, out of the board, and this is going back into the board. So we've got a chain that looks something like that. Oh, it didn't show up very well here. But I believe I've done 1, 2, 3, 4 as our parent, and we'll just let that be carbon 1.
And that's because it's the longest, right?
It's the longest, that's the parent. Now, we have 4 carbons, so that's a bute. It'll be a butane. You ask yourself, what's attached to your butane? We have 2 methyl groups, and they're both in carbon 2.
So it'd be 4.
Therefore,
butane.
it's a 2,2 dimethyl
Oh, wait, I can't get
butane.
Oh, Lord. Well, 2 and 2 equals 4, so, you know, I was there.
Now, what if I took this molecule here, and instead of having a hydrogen down here, what if I pulled one of these off and put a chlorine on? Let's do that. Actually, I put 2 chlorines on. It's still going to be a butane, isn't it? Now, what's attached to our butane?
2.
Well, we have 2 chlorines, don't we? So it's a dichloral. We still have 2 methyls attached. So it's a dimethyl dichloral. Chloral will come first. And where are we going to make our carbon number 1? Well, originally I put it up here, but here, if we made this carbon 1, there it comes, then we would have a 1 fluoro. That's our lowest number. So think about how you put this together then. We have 1,3-dichloro, 2,2-dimethyl, and it's still a butane. So that is systematic nomenclature.
Yeah, please. Wait. I don't have enough room.
All right, let's go backwards here. What if I give you a name and ask you to do a structure?
I will not get it right about now. Any of you guys?
Okay, 2,2,3-trimethylbutane. Step 1. It's a butane. So you need a zigzag with 4 carbons. That's where you go. You need a zigzag with 4 carbons. Bute is 4.
1, 2, 3.
And we'll call one end carbon 1. Doesn't matter which end. We just call one of them. Now what's attached to our butane? We have 3 methyl groups, right? And 2 of them are on carbon 2, and 1 is on carbon 3. So systematically, 2 methyl groups, 1 methyl group, and it looks like that.
I did that totally wrong.
I didn't even do it. I didn't even do it. Did I do it?
One more time. It's a butane, you draw a 4-carbon zigzag. Decide on which end you want to be carbon 1. Doesn't matter which. On carbon 2, you need 2 methyl groups. On carbon 3, you need 1. And there you are.
Okay, so number 2 should be 2 zigzags.
Carbon 2, 2 methyl groups. Carbon 3, 1 methyl group.
Wait, hexane.
Why is it called?
Okay, I think I got it.
I'm sorry.
Okay,
Why is it
hexane,
called a trimethyl?
6.
Because there are 3 of them.
So it should go
They're on different carbons, but there are 3 of them.
3, 4.
Show me the 3 of them on the structure.
1, 2, 3, 4, 5, 6.
1, 2,
right here.
3.
Okay, 2 now I get it. Thank you.
All right, our next guy is a hexane. So
6.
hex is 6.
Yeah.
Draw a 6-carbon zigzag.
I'm sorry, y'all.
And we'll let one end be carbon 1. Doesn't matter which end, let one of them be carbon 1.
That made me happy.
Now, according to our
I'm glad
name,
of that.
on carbon 3, we have an ethyl group. An ethyl, remember, is 2 carbons.
Oh, I'm gonna say 3. Oh, I was right.
So let's find carbon 3. That's 1, 2, 3 over here.
You gonna put 2,
You put 2.
And we put an ethyl.
1,
2 carbon side chain on carbon 3.
so 2 lines. 1, 2, 3, okay.
That's this.
2.
On carbon 2, we have 2 methyl groups. So carbon 2, 2 methyls, carbon 3, 1 ethyl.
1, 2. So, okay.
All right, this next one is the one that's on the test.
Oh.
What
It should be 8. So it should be 8
It's
zigzags.
8.
Oh.
8 zigzag. So you start off with a zigzag with 8 carbons.
Copy might be wrong now.
Wow.
We'll let one end be carbon 1.
So
Now.
2.
We have hex, that means 6 methyl groups.
Okay.
Where are they?
1, 2, 3,
They're on
4, 5,
carbons.
6,
2, 3, 4, 5, 6,
and 7.
and 7. Each of those has a methyl, and it looks like that. Now isn't that fun?
That's it?
Kind of getting it. Mm-hmm.
But this one will be on the test.
Right.
So just copy it.
Just copy it on your little note card.
I know.
All right,
Hey, Dr.
This
Young,
is just to
move
keep
that
me
too
honest.
fast.
Yeah.
can you go back real quick, please?
Sure.
Okay, that's it. That's all I needed.
Quick,
Thank you.
Okay. Okay. Just to keep me honest here. Remember I said that we have free rotation and that we regard these as all the same structure. In fact, they're not all the same.
I know I'm not ready.
Let's just look at a very simple molecule, and we're going to look at rotational isomers. So let's look at ethane. If you think about the single bond between two carbons, we could arrange the hydrogens two ways. This way is called eclipsed, where they're all exactly lined up. And this guy is called staggered. Just like in a sawhorse, we have one up, one down, and the others off to the side. These actually differ in energy. And you can appreciate that by looking at my little movie. As these hydrogens come around, see how close they get and there's a little fuzzy in between them? That means they're kind of touching. And that's higher energy. When there's perfect space between them, there's no touching. So as this bond rotates, the energy of the molecule changes. When they're close, that's the high energy. When they're in between, that's our low energy. These are conformational isomers. Now again, conformational isomers, we typically don't worry about. You can't separate these unless you're at very, 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 very low temperatures. But there's another type of isomer that we can. And these are called constitutional isomers. So now that we know how to name things, look at our first molecule. What's our longest chain here?
The second.
1, 2, 3, 4, 5 carbons, right?
Yes.
This also has, well, this has 1,
6.
2, 3, 4, 5, 6 carbons. But if you look at the molecular formula for each, what is it for this one? This is C7H16. Total of 7 carbons, 16
I'm
hydrogens.
about to cry.
Here we have 6 carbons in our main chain. There's number 7 sticking out. By gosh, they have exactly the same formula. But they are very clearly different compounds.
Are they both right though?
Well, they're both. Well, this is a 1, 2, 3, 4, 5. This is pentane. And this is a hexane. But they have the same formula. So they're isomeric. They're isomers, they have the same formula, but they're arranged differently. These are different compounds, and we can separate them. And that's one of the joys of organic, is separating things like this. One of the fun games that you can do with isomers is to give a problem like this. And this is write all the isomers having the formula C3H8. How many ways can you connect 3 carbons in a row and still have 8 hydrogens? How many ways?
Draw a ring.
3 points, that's it. You got 3 points. All you can do is that. 1 Z.
1, 2, 3.
So,
That's the only way you can do it. All right, what if I change my formula here from C3H8 to C3H6?
You draw 6. You draw 2 things, 2 zigzags.
Well, think about what you could do. How can we arrange 3 carbons and 6 hydrogens?
Oh, you draw 1 zigzag again. Right?
First of all, we can do a zigzag, but because we're missing 2 hydrogens, we need a double bond.
All right,
And
let's
what's
show
the
it.
other way? What if we take our 3 carbons and simply made a triangle?
See? I said that. Something like that.
Oh, boy.
A double bond. So here we have CH3, CH, and CH2. That's 6. CH2, 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 that's 6.
Oh, my God.
And these are isomers. They're different, but they have the same formula. Now, as we add more carbons, this can get more complicated. C4H8. How many ways can you arrange 4 carbons and 8 hydrogens?
I don't know.
We need a double bond somewhere, or a ring.
Draw a square.
A square. You got it.
Yo!
You got it.
Wow.
We have a double bond, a double bond, a double bond, a
That's
square.
funny, right?
We can take our triangle and put a CH3 off
I'm
the top.
complaining, y'all, for real. That was.
All of these are simple isomers. Same formula, different compounds.
We finna be drunk. Shapes. I'm having my daughter do this homework. Just come draw me some squares.
All right, one more here. This is the one that was on the exam. C3H6O2. Now remember, oxygen has a valence of 2, so we need 2 bonds to every oxygen. 4 bonds to every carbon.
You trying to stop something.
So go ahead and write quickly.
What's that, Dr. Young? Is that like ginger ale?
It's just water, sparkling water.
Oh.
Oh,
All right. How about these guys?
Oh, God.
C, 1, 2, 3,
Look, it wasn't like that, but
1,
it
2,
was
3,
close.
4, 5, 6 hydrogens, and 2 oxygens. Same thing with all these. But just to show you how interesting organic chemistry can become,
It is very interesting.
these are also isomers of the same structure. So we've changed our formula just a little bit by sticking in a couple oxygens, and we get a huge number of potential isomers. Organic chemistry can get very, very strange very, very quickly.
My head hurts now.
All right, what kind of questions do you have in an exam?
Every question.
Well, aside from this one.
Well, it is.
Look at these two. Are these identical compounds? Are they constitutional isomers? Or are they different and not isomers? Of course you know. Step 1. You want to look at your parent chain. They're both straight chains, aren't they? How many carbons in each chain?
5 on the first one. 1, 2, 3, 4.
5.
5.
We have 5 over here with 12 hydrogens. How many here?
5.
6.
1, 2, 3, 4. Oh, at the top, my
1, 2,
bad.
3, 4, 5, 6. These are different chemical compounds. Totally different. They don't have the same formula. They're totally different. Look at one more. A condensed formula and a structural drawing.
It's 8. No.
6.
So let's start here. How long is our main chain?
6.
1, 2, 3, 4, 5, 6 carbons. And we have 2 chlorines let's see, this chlorine is attached here, that would be carbon 3, so there's going to be carbon 2, 3, and 4. So we have chlorines in 2 and 4, and 6 carbons. Look at our next guy. What's our longest chain? 1, 2,
6.
3, 4, 5, 6. Okay, sounds promising, same thing. Where are the chlorines?
Meta, whatever you call it.
Dichloro. This would be in carbon 2, 3, 4. Carbon 2, and carbon 4. These are exactly the same, aren't they? Just one's a structural drawing, and the other is a line drawing. Any questions? All right, as we were looking at isomers, we were sticking in double bonds and stuff like that. Carbon can form single, double, and triple bonds. It still has to have 4 things bonded, but single, double, or triple. We call single bonds sp3. Double bonds will be sp2, and triple bonds will be sp. We'll see where that comes from in a minute. We'll also look at a couple more functional groups. Remember we said an alkane only had sp3 carbons. That's all. Well, an alkane, this guy with a double bond, is going to have 2 sp squared carbons, at least. An alkyne will have 2 sp carbons. So let's see where this comes from. Here we're going to take 1 s and 2 p orbitals. Again, that's where the nomenclature comes from. We're going to put this on each of the 2 carbons in our molecule. So here we're going to build a bonding network with 1 s and 2 p's, and we wind up with this. It's trigonal. Triangle. They're planar triangle. But remember, we have 3 p orbitals to work with, don't we? 3 of them. So we have 1 left over on each carbon. So the p orbitals are going to be on each of these carbons, and they're going to look like that. Now, when you take orbitals like this and line them up so that they're exactly lined up, they share electrons between the atoms, called overlap. And this forms what we refer to as a pi bond. Pi because they're made from p electrons. This looks like a hot dog. And there's the hot dog inside. There's the electrons around it. This is these 2 put together count as 1 bond. So we have 1 bond that's truly between the 2 carbons, and then we have the pi bond that's formed this way. That's what we mean by a double bond. 1 is our real bond, and the second one we draw is this pi overlap. Now this is really important because the pi overlap, this is a huge cloud of electrons. Chemistry of alkenes is going to involve that cloud of electrons and whatever. We'll see that in a second. Another neat thing about this, well, we'll see, here it is. You see, here's the big honking cloud of electrons. Remember I said that these have to line up. So if we took this and tried to rotate around this bond, we would wind up with these guys not lined up. So the bottom line here is you can't rotate. Once you form that pi bond, you're stuck with it. Now that's really important because both of these are alkenes. They both have 4 carbons. They both have 1 double bond. But in this guy, the methyl groups are on opposite sides. We call that trans. And on this side, they're on the same side. That's cis. They're totally different compounds because you can't rotate around this bond. Now, in fact, you can rotate about the bond if you put in energy. Everyone is familiar with the concept of vision, right? Do you know chemically what happens when your retina detects a photon of light? You start off with retinal that has a cis double bond. This double bond absorbs that photon of light, goes to an excited state, and when it does, it rotates to give a trans isomer. As it rotates, it tells your brain, hey, I just saw a photon. And that's how vision works. Isn't that neat? Because these have the same everything, but they differ only in space, they are called stereoisomers. One of the big things in organic chemistry is stereoisomerism. For an sp center, pretty much the same thing. We have one s and one p, and we build a linear structure like this. We have 2 p's left over on every carbon. These guys, py, pz, so they look like that. Once again, they overlap, and we form 2 sets of pi bonds. So one bond here is our single bond, one from this pi bond, and one from the other. Big cloud of electrons around this middle. So our 3 types of bonding in carbon, sp3, these are single bonds, sp2 are double, and sp are triple. We talk about these in terms of functional groups. Once again, we call this an alkane, an alkene, and an alkyne. Single bonds, at least 1 double bond, at least 1 triple bond. So let's go ahead and finish talking about functional groups. We said here we have a carbon-carbon double bond that makes us an alkene. If we have an OH attached on our chain somewhere, we call that an alcohol. If the oxygen is in between carbons, we call that an ether. Now there are lots and lots and lots of functional groups. And like I said, the neat thing about functional group chemistry is that regardless of the structure of, say, your alkene, the reactions are all going to be pretty much the same. So if you understand the reaction, the generic reaction, then you can take any molecule you have, any alkene, and predict the products. As an example, if we have an alkene and we add bromine, Br2, we will always wind up with a 1,2 dibromide, that means they're next to each other, and the bromines will always be on opposite sides. So regardless of the structure of your alkene, if you show a reaction with bromine, you know exactly what to write. The other thing we do in organic is to think about the mechanisms. This is a cycloalkene, double bond in a ring. This is bromine. This reaction goes stepwise. Step 1, the electrons in the ring are going to attack. That splits. and we form what we call a bromonium ion. Step 2, the Br minus that we clipped out here is going to attack the other side, break this ring, and we get our 1,2 dibromide from opposite faces. That's functional group reactivity. One of the things you do in organic is you look at functional group reactivity. This is partial set of reactions that alkanes undergo. And if you get to organic, the pleasure is you get to learn these things for dozens of functional groups. Really is neat. Let's go ahead and look at them. Again, we said alkanes, single bonds, alkene double, alkyne triple. If we take 3 double bonds and put them in a 6-carbon ring, we call it an arene. Here's all of our p bonds, our p orbitals. They overlap nicely and they make this amazing molecule we call benzene. Benzene used to be used widely as a solvent and stuff like that. When I was an undergraduate, we washed our hands with it, get the gunk off. It's now a carcinogen, so we can't do that anymore, but what can I say? Halide, we simply attach a halogen. Alcohol, we set an OH. Ether, the oxygen is in the middle. Attach an amino group. This is going to be important. Remember this. We attach a nitrogen here with a couple hydrogens. That's called an amine. We're going to use those when we do our intro to bio. Triple bond nitrogen, that's a nitrile. You can do a nitro group, an SH. SH tries to behave just like an OH, except it really, really stinks. The essence of skunk is butane thiol. So 5-carbon chain with an SH. The sulfide doesn't smell much better. Dimethyl sulfide is a horribly smelly compound. If we take a carbon and make a double bond, we call that a carbonyl. And we tend to put carbonyls in classes depending on what's attached to each side. If we have an H, it's an aldehyde. 2 carbons is a ketone. An OH on one side is a carboxylic acid. Again, this is going to be important. If we have an O with another carbon, that's an ester. This is very important to us for biochemistry. This is an amide. Carbonyl with nitrogen on one side, carbon on the other, halogen, or an anhydride. Carbonyl compounds form really the heart of organic chemistry because they are so very reactive. Remember we talked about electronegativity. Oxygen is more electronegative, so it tends to suck the electrons up this way. We wind up with a big red oxygen and kind of a blue carbon. That means that we can add things with electrons to this carbon. Let's pretend that we have this. Again, this is being pulled up. Oxygen can attack. Electrons go up, pick up a hydrogen, and we get addition. In this particular molecule, we could lose one of these hydrogens, kick it out as water, and we make an ester, a different functional group. Functional group organic chemistry is really the heart of organic, and by the time you do your 2 semesters of organic, you go through most of these functional groups that we've talked about. Lots and lots of reactions. Very interesting, very challenging. Well, I said that the amino group and the carboxylic acid were going to be important for biochemistry. Biochemistry is really just an outgrowth of organic. Amino acids. Think about the name. Amino acids are simply carbon chains with things attached. One of them is going to be the amino group, the NH2, and the other is the carboxylic acid functionality. Now there are 20 essential amino acids. They differ by what's attached here after our nitrogen. We go down our chain here. Here we have a guanidino group, it's called, but it's positive. Here we have, this is histidine with an imidazole group. Another amino group down here. These are the positively charged amino acids. At the end here, if we have a carboxylic acid, that's negative. Again, this one more carbon, but a negative group. These are our negative amino acids. We can also have uncharged side chains. So we have an OH group, an alcohol, another alcohol. This is an amide, another amide. This is an SH, a thiol. We can put selenium in there, believe it or not, or just a hydrogen. This guy is proline. This is what you use to make hair. Hydrophobic, that means nonpolar. Methyl group, isopropyl, isobutyl, another isobutyl, thioether. Here's a benzene ring. All of these guys are part of the essential 20 amino acids. Now in the body, what you do is you're going to take these amino acids and you're going to combine them and make the functional group we call an amide. It's a carbonyl, nitrogen on one side, something else on the other. Let's start off here with this simple amino acid. This is alanine, with just the methyl group as a side chain. Here's the amino group. Here's the carboxylic acid. This guy is a serine. It has an OH as our side chain. But again, the amino group and the carboxylic acid. In the body is capable of taking amino acids like this, reacting the amine with the carbonyl. Remember we saw a carbonyl get reacted with. Because this is positive, this has electrons. When that happens, we're going to split out HOH. That's water, isn't it? And when we do that, we form this bond. This is an amide, but in biochemistry, we call it a peptide bond. That's because biochemists, were biologists. They couldn't say amide, I don't know. But so this is a very simple peptide. It's simply two amino acids bonded together by the nitrogen. We can do this over and over again. Here we've stuck on a glycine and a leucine. This is called a peptide. A peptide is a series of amino acids. If it's a naturally occurring peptide, they will always be connected in a given sequence. We've all heard of growth hormone. Oh my goodness. Growth hormone is simply a peptide. Bovine growth hormone, by the way, is a different sequence than human, and so it doesn't work. So it's actually a bunch of bull. But what can I say? If we continue doing this over and over and over again, we wind up with a long chain that we call a protein. The primary structure of a protein is simply the specific sequence of all of the amino acids. Every time you make a specific protein, they're always lined up exactly the same. Now that's important because remember, each of these has this side chain, right? Some of them are water soluble, some are not. So as they are formed, they wind up into a ball. And they'll put all of the water solubles on the outside and all the hydrophobics on the inside. And you wind up with a tertiary structure of the protein. Depending upon the actual amino acids, see these little helices that can form? These are called alpha helix. The yellow up here is called a beta sheet. And here we just have the rest of the protein sequence going around. In an enzyme, this particular sequence is what, well, you see it will form crevices and things like that. In an enzyme, the substrate will bind to one of these crevices, and that will catalyze a particular reaction. Here this has gone to what's called cartoon format, where they accentuate the helices. In a second, there it goes, I hope. Here, this is the full space filling. Remember, blue is nitrogen. The charged compounds in the protein are on the outside, and the other stuff is on the inside. And you see the little crevices you can form. Substrates bind in those crevices, catalyze the reaction, and that's how an enzyme works. So what do you remember here? You remember that if you take amino acids, you make peptides by making amide linkages. You bind these together in a long chain, you make a protein. And the protein looks like this, constructed only of amino acids. Now we've also heard of RNA and DNA. How do they differ? Well, the R and the D simply stand for ribose or deoxyribose. Ribose is a 5-membered ring sugar. The difference is here on carbon 2, in deoxyribose, instead of an OH, you just have a hydrogen. So it's a very subtle difference between the two. In RNA and DNA, this will be our backbone, but we're going to attach what are called purines and pyrimidines. These are purines. They're 2 carbon rings with nitrogens, double bonds, and stuff like that. This is guanine and adenine. Again, they're attached down to the ribose. In RNA and DNA, these are going to be linked together by phosphate bonds. They're phosphate esters. So these are our purines. These are our pyrimidines. They are single ring, but again, heterocycle, nitrogens and carbons, and double bonds. Thymine occurs in DNA and RNA. We have uracil. Slightly different structure. All right. We talked about hydrogen bonding once. The particular structure of the RNA and DNA bases are such that when they line up next to each other quite properly, they can form hydrogen bonds. There's an NH and a carbonyl. Here's an NH and a nitrogen, and an NH and another carbonyl. The bases line up and hydrogen bond specifically. And that's how one strand of DNA knows what base has to be next to it, and that preserves the genetic code. If you look here, we have an A and a T, hydrogen bonded, 2 hydrogen bonds here. Here we have a GC with 3 hydrogen bonds, again, 2 hydrogen bonds, and again, 3. The backbone here is our ribose phosphate isomer with the bases attached, all hydrogen bonded very nicely. When they get together, they form something like this. And again, you can see the bases line up very specifically, hydrogen bond to each other. Our backbone here, once again, is going to be our ribose and phosphate. This will go through a couple renditions here. Let's wait and be patient. There you go. You can see all the negatively charged phosphates here on the outside, and our bases are buried here on the inside. That's actually important because water is so good at hydrogen bonding that if the bases were on the outside, they would never find each other. There's another rendition. Again, you can see now the bases, hydrogen bonding, and the phosphate all the way around the outside. All right, one more class of biomolecules I want to talk about, and these are lipids. We've all heard of fatty acids. We've heard of trans fatty acids and how bad they are. That's because they're bad. The body is built to metabolize 2 types of fatty acids. Fatty acid where we have no double bonds, so that's saturated is called, metabolizes that very fine, and cis fatty acids. If you have a trans fatty acid, the body cannot recognize it, cannot metabolize it, so it's bad for you. Saturated fatty acids are good for you. Unsaturated fatty acids are also good. Trans fatty acids are bad. If we take 3 fatty acids and hook them to a glycerol, that's an alcohol with 3 OHs, we form esters. This is saturated, unsaturated, saturated, called a glyceride, a triglyceride. Triglycerides are important. This end of the molecule is much more polar than this end. You can take this, convert it to a diglyceride like this, with a real heavy-duty polar end, so a phosphoric acid and an amino group, that's choline, and you get what's called a phospholipid. The neat things, okay, triglycerides, this is fat. This is how you would store fat. You convert it into triglycerides, move it into cells. When you want, or when you need energy, you simply cleave these off to fatty acids and burn them. The neat thing about phospholipids is that they form the barriers that surround every cell. That's the membrane. That's because this end is very, very water-soluble, hydrophilic, and this end is very insoluble, hydrophobic. They tend to auto-organize into something like this, where all the hydrophobic ends go together, and we form a bilayer. All of this is water-soluble, and this is not. This is the cell membrane. This is what separates the inside of the cell from the outside.
So semi-permeable when stuff are able to move inside the whatever the cell, like whatever you need to go in.
Well, to get anything...
Nutrients.
Yes, in order to get nutrients across the membrane,
I know.
we have to have very specific mechanisms. There are proteins that will bury themselves in the membrane, depending upon their side chains, bury themselves, called lipoproteins, and they will allow the movement of nutrients in and out of the cell. That's how that works. The other hydrophobic thing everyone's heard about is cholesterol. Cholesterol is a beautiful molecule. It's actually 3 6-membered rings fused to a 5-membered ring. We have another chain up here, and there's an alcohol here. This molecule, because of its particular stereochemistry, that's the orientation around all these bonds, see the wedges up and down, actually looks like this. It's very flat, very planar. Cholesterol is a very, very important component of membranes. There's lots of talk about cholesterol and diet and stuff like that. Turns out that your body makes it from scratch. Almost all the cholesterol that's circulating in your blood. Cholesterol that you eat has almost no impact. So your body needs it, it makes it. Cholesterol got a really bad rap in the 50s when President Eisenhower had multiple heart attacks, and they looked at his arteries and they were full of fat and cholesterol. And so they put the entire nation and most of the world on a low cholesterol, low fat diet, which actually has no scientific basis at all. No scientific basis at all.
I don't have my nothing. I don't have nothing.
So,
Because
all right,
is that why?
remember, next Monday,
I'll see if he's
we're
down.
going to have our sample exam. We're going to go over that.
Oh shoot! And this is the
Monday
last.
is our last day, right?
Yes.
So we'll meet over there somewhere.
Reviewing. Reviewing.
Yes.
Yeah, for the E3 review, probably downstairs where we were last week. And on Wednesday, all the way back here, we'll do our last exam. Now, at that time, if you have decided you want to take the final exam, please let me know then. Okay, the final exam, you don't have to take it. We can base your grade based on your exams. If you want to take it, let me know. Okay? Deal?
Deal.
Deal. All right. Have a good weekend.
You too.
So then technically next Wednesday is our last class.
That's it.
Okay.
Oh, come on. You're going to miss me.
We will.
That's true, though.
Not the class. We will miss you, but not the class.